Okay, did everybody get the email indicating that there were some study guide questions this semester that weren't applicable? Okay. If you didn't see that, you should look for it in your email. So let's start out seeing if you have any particular questions. Okay.
Peroxisomes?
Peroxisomes, we didn't really talk about peroxisomes. Don't worry about them. I'm sure you always like to hear that. Don't worry about it. Gap junctions, yes, we did talk about gap junctions. They were one of the three types of intracellular junctions. There were anybody remember what the three types of intracellular junctions were in animal cells? Tight junctions, desmosomes, and gap junctions. Okay, what do tight junctions do? They stick cells together that are adjacent to one another. Remember the intestine, you don't want materials leaking between cells. And so they kind of join the cell membranes of two adjacent cells together, kind of like little spot welds. What do desmosomes do? They also anchor adjacent cells, but those cells are ones that are subject to a lot of mechanical stress. And you don't want that tissue to pull apart. And so the desmosomes are a whole bunch of kind of fibrous proteins that are in the membranes, and then they actually extend into the cytoplasm and anchor to the cytoskeleton, and that provides a lot of internal support for those cells so that they don't pull apart. And then gap junctions are the protein tubules that join the cytoplasm of two cells, and this allows cells to communicate with one another. Remember the example I gave in animal cells? Some tissue that needs to communicate and how they communicate? Cardiac cells. When the muscle cells in your heart contract, after the initial stimulation by the pacemaker, which is electrical, then that wave of contraction is propagated by the flow of ions from one cell to another. And those pass through those gap junctions, and that's what stimulates subsequent contraction of those particular cells. So those were the three animal junctions. What was the only plant junction we talked about? The plasmodesmata, okay? And that's actually cytoplasmic connections between cells. So cytoplasm actually traverses the cell walls of adjacent cells. And remember, that allows plant cells to communicate. I mentioned that plant hormones, for example, can move through those plasmodesmata, and that can help to regulate plant cell growth, which is how plant cells respond to changes in their environment. I guess a long answer to a short question. Surely there are other questions. You already know it all, huh? Or, alternative.
Aquaporins, and when are they used instead of just like general osmosis?
Okay, we didn't talk about aquaporins, but aquaporins are actually channels through which water can move. If you think about it, when we talked about permeability of membranes, we said that membranes aren't permeable to polar molecules, and yet water's a polar molecule, but membranes are very permeable to water, and that's because of the presence of these channels, these aquaporins, but I didn't use that term. Classic example of that was in the textbook, right?
Yeah, Carl wrote it like that.
Okay, something that was in the textbook I didn't talk about, not responsible for it on this exam. Well, maybe this will stimulate some other questions. Let's try this. This is last year's exam. which isn't one of the ones that was posted. And so if there are not questions right now, what we could do is try going through some of the questions on this exam and kind of reasoning through them so you can kind of see what the questions look like and maybe get an idea of kind of a process you can go through as you address these particular multiple choice questions. However, as we're going through these, if additional things come up that you're uncertain about, please ask, okay? So, read question number 1. Which of the following is false about dissociation? Give yourself a second to just pick out what you think is the best answer. What I really like to do, and I tried to when I wrote the exam, is to underline when we have false STEM references. So basically this ends up being a true-false question. And so what I like to do when I go through these is, for each of the choices, just put a little T or an F by it so you don't get mixed up in terms of answering that particular question. Okay, so does dissociation explain why water molecules hydrogen bond to one another? True or false? That's false, okay? At least tentatively, you can see if there's a worse answer, a more false answer. Why do water molecules hydrogen bond to one another? What feature of water explains why hydrogen bond to one another, water molecules? The fact that water's polar, right? And the fact that there's a transient charge where oxygens are negative charged on one water molecule, hydrogens on another water molecule are positively charged, and that weak attraction between two water molecules is a hydrogen bond. Okay? Does water spontaneously dissociate? True or false? That is true, right? It spontaneously dissociates into what? H pluses and OH minuses. So we've got a T for that one. Does it explain why table salt dissolves in water? Yeah, it does. Okay, sodium and chlorine are ionically bonded, but when you put them in a water solution, they dissociate. They dissolve. So that's a truth. Do water molecules dissociate at a very slow rate? Yeah, they do. Okay, only a tiny amount of water molecules, I think I said 1 out of every 554 million is dissociated at any given time. That's why those concentration of hydrogen ions is so small. At equilibrium, what's the concentration of hydrogen ions in a pure water solution? 1 times 10 to the minus 7th molar. And that translates into a pH of what? pH of 7, because pH is what? The negative log of the hydrogen ion concentration. So the minus log of 1 times 10 to the minus 7th, the number of hydrogen ions that are produced by dissociation, is 7. Okay? Okay, question number 2. Water is an essential molecule for life due to its many unique properties. This feature of water best describes why that's the case. The latter 3 right down here are features of water. Okay? These are some of the essential features that makes water essential to life. But they're not the cause of that. Are water molecules joined by ionic bonds? Water atoms are joined together by polar covalent bonds. And because it's polar covalently, then water has these unique properties, such as high heat of vaporization, being less dense as a solid, water serving as a buffer. Which of the following about the pH scale is false? Another kind of true-false question. True or false? Is it a logarithmic scale? That is true. Okay. If a solution has a pH below 7, does it have more hydroxyl ions than H plus ions? Below 7. It's acidic, right? So it's got more H pluses than OHs. That's false. Now don't just stop there. Okay, always read your other choices, because there could be a B and C, for example, type choice. Okay? What about this statement? The H plus concentration is higher in acidic than a basic solution. True or false? That's true, right? Okay. Does the scale range from 0 to 14? Yes, it does. Okay. Question number 4. Even if acid is added to the blood, this prevents hemoglobin from changing to a shape that won't effectively bind oxygen. What about dissociation? Is that relevant to that question? No. How about the bicarbonate and carbonic acid in the blood? Bicarbonate and carbonic acid, a weak acid and a salt of a weak acid, make up a buffering system. Buffering systems are what are responsible for proteins maintaining their shape in a solution like blood, even if we add acid or base to them. Okay? Does the fact that water has a high heat of vaporization or that it's cohesive have anything to do with change shape of hemoglobin? No. Those are properties of water. So the correct answer here is B. Buffers resist pH changes when acid or base is added to a solution. And the reason hemoglobin might change shape, denature, is because the environment might become too acidic or too basic. Question number 5. Definition. What's the pH of a solution with an H plus concentration of 10 to the minus 6 molar? 6. Okay. Question about carbon. Can carbon bond with carbons and hydrogens to form hydrocarbon chains? Yep, that's what a hydrocarbon chain is. Can these chains be branch or linear? Yep. Can they form ring structures? Yep. Does carbon have a valence of 4? Yes, it does. Okay. If you look at carbon up here, atomic number 2. Remember that first orbital holds how many electrons maximum? 2. So that one's full. Next orbital holds how many? Up to 8. And there's 4 remaining of those 6. So it's half full. It needs 4 additional electrons to complete its outermost octet to be stable. Its valence is 4. Okay, dehydration reactions. What's the opposite of a dehydration reaction? Hydrolysis, okay? So don't confuse hydrolysis and dehydration. So which one of these is applied? Hydrolysis reactions split large molecules into their monomers. That's what the lysis referred to as splitting. And what does the hydro refer to? Water molecules are consumed in that process. Okay, what do dehydration reactions do? They synthesize, they build up monomers into polymers. Okay? And that liberates water molecules. Okay? So, do they break up polymers into their monomers? No, that's hydrolysis. Do they cause water molecules to hydrogen bond? No. Do they produce water as a byproduct? Okay. Do they consume water molecules? No, that's hydrolysis. Do they change the valence of atoms involved in the reaction? No. Valence is a property of matter, and those reactions won't change it. If you really didn't know, what strategy could you use? You might look at these questions and say, well, these are kind of directly opposed to one another. So it's probably one of those two. Okay, major difference between steroid hormones, testosterone, and estradiol, or estrogen, is now here's where reviewing your PowerPoint slides is really important, because this was from one of the PowerPoint slides that I showed last year, this year, I'll show it next year. And that is the male and female lines and the structures of testosterone and estradiol. And I pointed out very specifically on that there's a very small difference in the structure of those particular hormones, those sex hormones. And it's a couple of functional groups. Okay? So the correct answer here is basically two different functional groups. They both have covalent bonds. Estradiol has double bonds in it. These molecules don't have pH. Okay, pH refers to hydrogen ion concentration. Okay, I said you needed to be able to recognize functional groups, and so there was one question that had to do with recognizing functional groups. What's this functional group right here? Amino. Sulfhydryl. Not a carboxyl, is it? What do you need down here? You need an OH for that to be a carboxyl. So this is a carbonyl functional group. There are two kinds of carbonyl functional groups, aldehydes and ketones. Which one is this? This is an aldehyde because it's at the end of the chain. It would have to be in the middle of the chain if it were a ketone. What's this? Phosphate hydroxyl. Okay. So this one asks which one contained an aldehyde functional group. That would be C. So, what did Hooke and von Leeuwenhoek do? Remember, we said that he's kind of considered to be the father of microscopy and developed the first microscopes. Remember what Hooke did? Cork tissue sections, and he coined the term cell. Okay? They were not, though, responsible for proposing the cell theory. I think I said, it's kind of hard to know who to credit toward actually developing the cell theory. Probably if you ask people, they would say Schleiden and Schwann. Those aren't terms or people that I told you. You don't have to remember that for the exam. These were well before the first electron microscopes. Okay, the electron microscopes were developed in the 1940s and 50s. These guys were back in the 1600s. They didn't develop cell fractionation techniques, and they have nothing to do with models of membrane structure. Okay? First model of membrane structure didn't come until the late 1800s. Okay, which of the following is a type of electron microscopy? There's two major categories of microscopy: light microscopy and electron microscopy. How do they differ? Differs on what energy source you use to view a specimen. In light microscopy, you use light as a source of energy. In electron microscopy, you use electrons. Okay? So what are light microscopy? This is light microscopy. This is scanning and transmission are the two major categories of electron microscopy. Okay, fairly long question. If you homogenize tissues and then take that material and subject to differential centrifugation, and then analyze the pellets at different speeds, what question would you most likely be investigating? Okay, so think about it. You take some tissue and you bust it up. And you release the cells and all their cell organelles and things. And then you start spinning it. First slow, collect a pellet, then take that supernate and spin it again, collect another pellet. So basically you're collecting organelle fractions. Okay? So probably what you would be doing is trying to figure out in which of those fractions is this process of glycosylation taking place. Okay? Does the tissue consist of prokaryotic or eukaryotic cells? No, you're looking at cell organelles. Okay? Is the cell theory valid? Well, to look at that, you'd have to look at all kinds of different organisms and see if they were made of cells. You can do that with the microscope. You don't have to do centrifugation. It has nothing to do with pH and biological molecules. And I don't know how you would ever think that it would be that answer was von Leeuwenhoek, the father of microscopy. Okay, this cell structure must be present in both prokaryotic and eukaryotic cells. Remember, we said there were some minimum requirements for anything to be a cell. Is that a requirement to be a cell? Nope, you've got to have genetic information, but it doesn't have to be enclosed in a membrane-bound nucleus. You have to have ER. Nope. You have to have mitochondria. You have to have chloroplasts. But you got to have ribosomes, right? Okay, cell that has a very high surface area to volume ratio. What happens to your surface area to volume ratio as cells get bigger and bigger and bigger? Surface area to volume ratio decreases, right? Because one's increasing by the square of the size increase, one's increasing by the cube. So the volume's getting bigger relative to the surface area. Okay? So would it typically be one of the largest size cells? No, it'd probably be a small cell if it had a good surface area to volume ratio. Would it have a high metabolic rate? If you've got a lot of surface area, then you can transport things into the cytoplasm and get rid of waste very effectively. And that's necessary for a high metabolic rate. So that's a good possibility for a candidate for a correct answer. Generally speaking, smaller organisms have higher metabolic rates because of that surface area to volume consideration. The higher your metabolic rate, the faster you can reproduce. The larger you are, probably the slower you would reproduce. Okay? Whether you're a plant cell or an animal cell probably isn't really relevant to that. So the best answer here would likely have a high metabolic rate. Endomembrane system. Basically, which one of these is not part of the endomembrane system? Golgi, yes or no? Yes. Lysosomes? Lysosomes, yes, because remember that vesicles pinching off of the Golgi can fuse together to form a lysosome, so it's part of the system. Vesicles? Yep. Mitochondria? No. Okay. Where do we think mitochondria came from and chloroplasts? Bacteria that basically invaded or established a relationship with the first eukaryotic cells. Do you remember what we called that theory that explains that? Endosymbiosis. Because turgid plant cells have a large central vacuole filled with water, it's essential that they also have this cell structure. What does turgid mean? Swollen. Okay? Why is this cell swollen? Because its vacuole is filled with water and pushing it out. What's a turgid cell going to tend to do? Pop, right? Lyse? Unless it has what? Unless it has a cell wall. Under what conditions would you expect to find the cell with the predominance of membrane-bound ribosomes? So this question is asking you to distinguish between the function of membrane-bound ribosomes and free ribosomes. What do membrane-bound ribosomes do? They produce proteins that are secreted. Okay? So a cell that is secreting proteins would tend to have a lot of membrane-bound ribosomes. What if you were mainly producing cytoplasmic enzymes? No, you're not secreting them. They're staying inside the cell. What about a cell that's getting ready to divide? Well, if a cell's getting ready to divide, it's got to make new things for the daughter cells. Those are things that are going to stay inside the cell, like cytoplasmic enzymes. And this really isn't relevant at all, a cell that's digesting food particles. So, secretion, membrane-bound proteins. Okay. Everybody should be able to answer the next one, hopefully. If you were listening a minute ago. That's the correct answer. Okay. Dynein walking is a key event in this cellular process. Correct answer. Chemiosmosis refers to using a hydrogen ion gradient to make ATP. It has nothing to do with Dynein walking. Amoeboid movement? Nope. That has to do with the smaller elements of the cytoskeleton. Dynein walking is associated with the microtubules. Remember the microtubules sliding past one another? As a result of this, Dynein walking causes cilia and flagella to rotate back and forth. So motility using flagella. Structure of this cell organelle is directly tied to its function and involves having an inner membrane which separates two compartments. The two compartments are the matrix and the inner membrane space. Which one? That is the mitochondria. Remember our room has the mitochondria example. Here's our inner membrane space. Here is the inner membrane, and here is the matrix. And in the mitochondria, we're going to build up a hydrogen ion gradient. High here, low there, and use that as the source of energy to power ATP production by chemiosmosis. Okay, a defective receptor in a protein that binds LDL is responsible for does it cause a loss of function of the sodium-potassium ATPase? No, not related. Familial hypercholesterolemia. Remember that elevated cholesterol levels, okay? LDL is the low-density lipoprotein. Remember what Tay-Sachs is? What context I mentioned that in? I mentioned that you can have some genetic diseases called lysosomal storage diseases, where some of those enzymes in the lysosome don't work properly. Tay-Sachs is a lysosomal storage disease. Has nothing to do with disruption of facilitated diffusion. Membrane proteins play all these roles in the cell except correct answer. Do proteins have enzymatic activity? Membrane proteins? Yep. Sodium, potassium, ATPase, right? Are they transporters? Absolutely. Some of them are. Do they play a role in cell signaling? Remember I mentioned that receptors can transmit signals from the outside to the inside. Talked about insulin. Cell recognition. White blood cells determining whether it's foreign or self. So the correct answer would be, they all play a role. This type of transport would be used to move a polar molecule across a cell membrane from its high concentration to its low concentration. Which two can we eliminate immediately? D and E. Why? Because we're talking about movement of a molecule. What do these involve? Bulk transport. Okay? Cells or big chunks. Okay? So what are the key here? It's polar. So is the membrane permeable? Nope. So what does that rule out? Rules out diffusion. We're moving from a high concentration to a low concentration. What does that rule out? Rules out active transport. So facilitated diffusion is the choice. In class, we concentrated on the role that the sodium potassium ATPase membrane protein plays in the function of this cell type. I didn't talk about helper cells this semester. If that doesn't sound familiar to you. I did mention B lymphocytes. Anybody know what a B lymphocyte does? They produce and secrete antibody. There's the antibody-producing cells. Conformational change in a membrane protein is most likely involved in this membrane transport process. You're really only down to two choices, okay? Because again, these are all bulk transport processes, and those don't involve conformational changes in individual membrane proteins. So you're really down to simple diffusion and facilitated diffusion. Why do we need a carrier protein? Because the membrane's not permeable to the molecule, right? Is permeability an issue in simple diffusion? No. Membranes are permeable to substances that diffuse. But in facilitated diffusion, we need this carrier to move substance across the membrane because it isn't permeable. And conformational change is likely a mechanism involving that carrier. Okay, another fairly lengthy question. So a normal solute concentration of a cytoplasm of red blood cell is 0.7%. If you do an emergency transfusion of pure water, which has no solute, or 100% water, what is this water concentration inside the cell? 99.3%, right? So what's going to happen as a result of osmosis? Water is going to go from its high concentration to its low concentration. So the cell will swell and burst because it has been placed in a hypotonic environment. The cell is hypertonic compared to the transfused water. Okay? So the correct choice here is they would burst because they were placed in a hypotonic environment. Roadside plants may be killed because salt accumulation in the soil. How come? Does this choice make sense? Well, will adding salt to the soil make that solution hypertonic, more salty? Yeah. And what's that going to do? That's going to pull water out of the plant roots. Okay? Would the plant roots swell and burst if the soil was too salty? No. You really don't have any evidence to evaluate this, or this, or this. Okay? I suppose they're possible, but this is the best choice because you've got some evidence that this makes sense. And again, these two things are kind of opposite from one another. And so they're likely are the two that you could be concentrating on. Which of the following is inconsistent with the currently accepted model of membrane structure, the fluid mosaic model? Do phospholipids tend to flip-flop? Remember what flip-flop means now. Moving from one side of the bilayer to the other. Very little, right? Remember that phospholipid bilayer? Flip-flop involves this one going over here. And that hydrophilic head would have to rotate through that hydrophobic core of the membrane. Not going to happen very often. Will some membrane proteins freely move within the phospholipid bilayer? Yep. Are some restricted to a particular location? If they're anchored to the cytoskeleton, they would be. Okay. Is cholesterol present in the phospholipid bilayer? Yep. So flip-flopping rarely happens. In sucrose co-transport, the active transport of sucrose into plant cells is coupled to a proton pump, but the proton pump requires ATP hydrolysis. So these both are correct answers for this particular question for sucrose co-transport. Okay, we've got an animal cell exposed to a lower temperature than normal for a long period of time. What would be the most likely consequence? What is lowering temperature going to do to fluidity? It's going to make the membrane more viscous. It's going to decrease fluidity. Okay? It wouldn't become more fluid. It'd become less fluid. Might it respond by increasing the amount of phospholipids with unsaturated fatty acids? Yep, because remember with those unsaturated fatty acids have the double bonds in the fatty acid chains, and they cause those kinks in the molecule, and that keeps the phospholipids from packing too tight together when the temperature decreases. So that helps to compensate for colder temperatures. Would it increase the protein content of its cell membrane? That would make the membrane more viscous. More big molecules in the membrane would keep the phospholipids from moving around. Would you start drinking from water supplies that were more salty? Not really relevant. Would it respond by increasing the amount of smooth ER in its cells? Not really relevant. Model of membrane structure, phospholipid bilayer sandwiched between two layers of protein stuck on the inner and outer surfaces. Cell theory, so they're eliminated. Fluid mosaic model, that is the Davson and Danelli model, that is the phospholipid sandwich. Okay, some agree and disagree questions. So here's the statement. The fact that water has polar covalent bonds helps to explain why hydrogen molecules bond with one another. So your first decision is, will I agree with it or disagree with it? That is a pretty easy statement to agree with. Okay? Because water molecules have polar covalent bonds, remember a polar covalent bond results in unequal sharing of electrons between the atoms. So they spend more time around the oxygen than they do the hydrogen. And this is the hydrogen bond that joins water molecules to one another. So that would be a very easy explanation to make. Now, again, if you're really stuck with this, start identifying some terms. Okay? So tell me what a hydrogen bond is, or tell me what a polar covalent bond is. Okay? Antibiotics exploit differences in prokaryotic eukaryotic cell structures to selectively kill bacteria while not harming the eukaryotic host body cell. What would be an easy way to agree with this statement? What's the differences they exploit to be selective? 70S versus 80S ribosomes, right? However, I could see you disagreeing with this statement too, in the following fashion. Not all antibiotics inhibit protein synthesis. There's another class of antibiotics that do what? Remember how the penicillins work? Cell walls. They destroy the bacterial cell wall. So you could disagree with it and say, well, some antibiotics do that. The protein synthesis inhibiting antibiotics. But some, well, I guess you still wouldn't be disagreeing with it. You'd just be explaining in that particular case the difference being cell wall versus no cell wall, 70S versus 80S ribosome. So yeah, it'd be hard to disagree with this statement. There could be different ways to agree with it. We didn't talk about this. We didn't talk about humoral immunity and cell-mediated immunity, so don't worry about that one. And there are at least 2 conformational changes that must occur within the sodium potassium ATPase membrane protein in order for it to carry out its intended function. What is its function? Remember? Pump 3 sodium out, 2 potassium in. Does that require conformational changes? You bet, a whole bunch of them. And if you could describe 2 of them to me, you'd be in good shape. Okay, first essay question asked last year had to do with lysosomes. So remember intercellular digestion. It's got those digestive enzymes inside. Remember the term autophagy? Does that name strike a bell? That's where a lysosome can be used to recycle old worn out cell organelles, like an old mitochondria. We didn't talk about this time. What's the adaptive advantage of having H pluses? Of this being more acidic? And in the last essay question, dealt with the difference between facilitated diffusion and simple diffusion. How are carriers involved? How are conformational changes involved in facilitated diffusion? How does active transport differ from facilitated diffusion? How are they similar? What about that last question? How does active transport differ from facilitated?
Active goes against the gradient.
Active goes against the concentration gradient. Facilitated goes with it, down it. It's another difference.
Active transport uses ATP.
Yep, active transport requires ATP because you're going uphill, if you like. How are they similar? They both need a protein carrier because the membrane's not permeable to those substances. So I would recommend that as you get your notes mastered, that you then go through the 2 posted exams in a similar fashion. And don't, I almost took them off there. I've got the answers on there. Don't just go there and say, yeah, that answer's right, I understand that. Go through each of the choices and try to make sure you understand not only why the correct answer is correct, but what's wrong with the incorrect choices. Because that'll help you to learn some of the stuff. So we've got 2 minutes. Any last minute questions? Sorry?
No lab.
Oh, yes. No lab quiz, pre-lab quiz next week. Okay? Because we knew that Monday's lab would just be trash anyway, so you're not going to be thinking about that. So no pre-lab quiz, and obviously no reading quiz. Okay? If you want to do the optional points, remember this afternoon 3:30 is 1 physics person. Monday, I forget what time I told you, and Wednesday. One other thing that you have?
Yeah, just because there isn't a lab quiz doesn't mean it isn't important to read the lab ahead of time.
True, true. You still need to read the lab ahead of time. You're just not going to get quizzed on it. Okay, so get here as quickly as you can on Monday. Okay? You've got maximum amount of time.
I'm just gonna write down all the equations. Yep.